Now we come to the tryptophan operon. What is tryptophan operon? It has 5 structural genes. If you watch my previous video, I showed you that there were 3 structural genes in the lag operon region. These were this thing like Z, like Y, like A. Similarly, tryptophan has 5 structural genes that code for the enzyme of the synthesis of amino acid tryptophan. So the messenger RNA that is actually produced from these structural genes is going to code for the amino acid tryptophan. But what happens now is that the tryptophan operon or its position is under negative control. So what happens is, for example, this was that this was the site where the repressor gets bound, the operator site. So what happens is that tryptophan binds to another substance, acts as a co-repressor and gets bound to this operator site and acts as a weak repressor. What is this weak repressor going to do? It is going to attenuate. So this weak repressor is going to form a messenger RNA, but this messenger RNA is going to form a hairpin-like structure. This hairpin-like structure, we studied that in Rho-independent termination as well. What is this structural hairpin-like gene is going to do? This hairpin-like structure is going to stop the further synthesis of tryptophan in case of tryptophan axis. So now, as we know, this is what happens in prokaryotes. So we know that prokaryotes are having transcription and translation side by side. So this messenger RNA, which is not going to code for anything, this messenger RNA is going to be translated into some substance, which over here they have written is a truncated non-functional peptide product. And this peptide product is degraded. So this tryptophan acts as a weak repressor and eliminates the tryptophan production by the cause of attenuation. Now, if tryptophan is less, if tryptophan becomes scarce, the operon is expressed. What happens is the 5' band of messenger RNA contains 2 adjacent codons for TRP, and the lack of the tryptophan causes the ribosomes to stall at these 2 codons. So here and here 2 ribosomes will get attached, and when they will get attached, no messenger RNA hairpin loop-like structure will be formed, and the tryptophan messenger RNA will be formed, which is converted by the nearby ribosomes into their proteins. So this is how it actually happens. This is the ribosome, and the ribosome, when it's attached, it is going to prevent the formation of the hairpin loop structure. And this hairpin loop, the absence of this hairpin loop will actually help the tryptophan to be formed. So the negative regulation was done by attenuation, whereas the positive regulation is done by the ribosomes to stall at the codons. And this prevents the attenuation program and allows the transcription to be continued. And this is how tryptophan regulation is done. I hope you understood this. If you have understood this, we'll go to the coordination of transcription and translation next.